Right, we start out with bioenergetics and basic principles. The quantitative study of energy transductions in living cells, and also study of the nature and functions of the chemical processes during the reaction. Two fundamental laws of thermodynamics. First law is the principle of the conservation of energy. For any physical or chemical change, the total amount of energy in the universe remains constant. Energy may change form, or it may be transported from one region to another, but it cannot be created or destroyed. The second law of thermodynamics. In all natural processes, the entropy of the universe increases. The three thermodynamic quantities, universe is equal to the reacting system plus surrounding environment. Free energy, G. It is the amount of energy capable of doing work during a reaction at constant temperature and pressure. The free energy change is denoted by Ag. In hexagonic reaction, there is a release of free energy, delta G is negative. In antagonic reaction, gain of free energy is delta G is positive. Right? Now these are all PowerPoint notes. Make a good screenshot of each so you could use it for future references. The three thermodynamic quantities. Enthalpy, H. The heat content of the reacting system. It reflects the number and kinds of chemical bonds, covalent and non-covalent, in the reactants and products. Releases heat, exothermic. The change in enthalpy, delta H, is equal to a negative value. Take up the energy, endothermic. The change in enthalpy, delta H, is a positive value. The three thermodynamic quantities. Entropy S, quantitative expression for the randomness or disorder in a system. When the products of a reaction system are less complex and more disordered than the reactants, the reactant is said to proceed with a gain in entropy. The units of delta G and delta H are joules per mole or calories per mole. The unit of delta S is joules per mole, and so Kelvin, that's joules per mole asterisk Kelvin. 1 calorie is equal to 4.184 joules. The gas constant R is equal to 8.315 joules per mole Kelvin. Is equal to 1.987 calories per mole Kelvin. Units of absolute temperature, T are Kelvin, K, 25 degrees centigrade, is equal to 298 Kelvin. At 25 degrees centigrade, RT, right? I hope you know what RT is. I expect you to find it out. All right? If you have any doubts on that, post a comment, I'll give you a reply. 2.478 kilojoules per mole is equal to 0.592 kilocalories per mole. Right? The relation between delta G, delta H, and delta S. The changes in free energy, enthalpy, and entropy are related to each other quantitatively by the equation delta G is equal to delta H minus T delta S. Equilibrium constant. The concentrations of reactants and products at equilibrium define the equilibrium constant, KEQ, small a, capital A plus small b in a capital B is reversibly equal to small c, capital C plus D, capital D, where A, B, C, and D are the number of molecules of A, B, C, and D participating. The equilibrium constant is given by KEQ is equal to CEQ, TEQ by AEQ, BEQ, with the suffixes of small c, D, A, and B, right? Where A, EQ, BEQ, CEQ, and DEQ are the molar concentrations of the reaction components of the points of equilibrium. Next will be free energy change in a reaction is expressed by delta GP is equal to delta GT infinity plus RT ln products by reactants. Equilibrium constant and standard concept of free energy. Transform constants as standard free energy changes and standard equilibrium constant is delta G is equal to delta G0 is equal to RT link ln gas constant, and table relationships between KEQ, delta G, and the directions of chemical reactions. When KEQ is, delta G is, and starting with all components at 1 m, the reaction greater than 1, we have negative, it proceeds forward. At 1, it's 0, is at equilibrium. Less than 1 is positive, proceeds in the reverse. Equilibrium constant and standard concept of free energy. When delta G is large and negative, the reaction tends to go in the forward direction. When delta G is large and positive, the reaction tends to go in the reverse direction. And when delta G is equal to 0, the system is at equilibrium. The free energy change for a reaction is independent of the pathway by which the reaction occurs. Metabolic pathways. These are all the configurations. Exactly. In fact, they are representations of the glycan biosynthesis and metabolism, nucleotide metabolism, biodegradation, xenobiotics, carbohydrate metabolism, lipid metabolism, amino acid metabolism, metabolism of other amino acids, metabolism of cofactors and vitamins, biosynthesis of secondary metabolites, energy metabolism. And here you have the cell macromolecules, proteins, polysaccharides, lipids, and nucleic acids. And the energy containing nutrients, carbohydrates, fats, and proteins. So during the process of catabolism, ADP plus HPO3, NAD plus, NADP plus, and FAD plus undergoes catabolism to give ADP, NADH, NADPH, and FADH2. And energy is depleted, and energy depleted products CO2, water, and ammonia. And when it undergoes anabolism, it produces ADP, HPO3, NAD plus, NADP plus, and FAD. So we start off, it goes from the energy containing nutrients, they undergo catabolism to release this, and then undergo anabolism to release these, to get deducted, and we have cell macromolecules that are also involved. And the precursor molecules are amino acids, sugars, fatty acids, and nitrogenous. Bases. The big picture energy relationship between catabolic and anabolic pathways is what is shown above. And this is a flow chart of the cycle pathway, the citric acid, right? Or the TCA cycle, acetyl acetyl-CoA 2 on these being formed and then being released this way. Phospholipids and triacyl glycerols, starch, glycogen, sucrose, they all in the presence of alanine, glucose, and serine. And here the phospholipid triacylglycerols and go into fatty acids, and if you add here phenylalanine, pyruvate, leucine, and isoleucine, they get converted to acetyl-CoA, which then gives acetoacetyl-CoA, and to one form it releases malonate, isopentyl pyrophosphate, rubber, keratin pigments, etc. And you can see the diverging anabolism, the cyclic pathway, right? These are bioenergetics. Right, now going to the bioenergetics. Glucose occupies a central position in the metabolism of plants, animals, and many microorganisms. Rich in potential, energy, oxidation of glucose results in standard free energy change of minus 2,840 kilojoules per mole. Remarkably versatile precursor capable of supplying a huge array of metabolic intermediates for biosynthetic reactions. Extracellular matrix and cell wall polysaccharides, glycogen, starch, and sucrose. Synthesis of structured polymers, storage. So glucose gives rise to all these. Glucose also gives ribose phosphate and pyruvate. In the presence of oxidation by pentose phosphate pathway. In the presence of oxidation by glucose, it gives pyruvate. In the presence of storage, it gives glucose, starch, sucrose. In the synthesis of structural polymers, glucose gives extracellular matrix and cell wall polysaccharides. Bioenergetics and glycolysis pathway. Metabolites like glucose are often activated with a high energy group before catabolism. Glycolysis in 10 steps. In glycolysis, 2 molecules of the 3-carbon 3C compound pyruvate first metabolic pathway to be elucidated by Edward Buckner's in 1897. The glycolytic pathway, glucose is the sole source of metabolic energy in some mammalian tissues and cell types, erythrocytes, renal, medulla, brain, sperm. Glycolysis has 2 phases here. In preparatory phase, 1 to 5th step, investment of energy. And the second payoff phase of glycolysis, 6th to 10th step, gain of energy. And the next we have is the 14th PowerPoint presentation. Preparatory phase, phosphorylation of glucose and its conversion to glyceraldehyde triphosphate. And it gives these, which things we have already discussed. It's just giving you the illustrations of these. So take a note of when glucose in the presence of hexokinase and then gives glucose 6-phosphate, which again in the presence of phosphohexose isomerase gives you fructose 6-phosphate. And here in both cases here ATP is reduced to ADP, and here again in the 3rd step ATP becomes ADP. Then it gets this fructokinase helps to convert fructose 6-phosphate to fructose 1,6-biphosphate in the presence of aldolase, and then it has a reversible conversion to glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate, and then in the case of triose phosphate isomerase. All right, we have the formation of glyceraldehyde 3-phosphate, which is the payoff phase, oxidative conversion of glyceraldehyde 3-phosphate, pyruvate, and the coupled formation of ADP and ADH. So the entire thing is an illustrative lesson today. So make a note of each screenshot and study them, okay, constantly, and you should be okay, right? And that's it about that depiction there. Then this is the next one for I'm just magnifying it for you. All the steps, the enzymes, the cofactors, end products, the free energy, and the EC and okay, we'll have to figure that out, and others also. It's okay, and we have a balance sheet which is glucose plus 2 ADP plus 2 NADH plus 4 ADP plus 2 pi gives 2 pyruvates plus 2 ADP plus 2 NADH plus 2 H2 plus 4 ADP plus 2 H2O. These are all formulas you can always remember for the exams. Cancelling out common terms on both sides. See, if you can have common terms, you cancel out 2 ATP, take 2, you have another 2 ATP here. Likewise, you cancel out both terms, finally you get glucose plus 2 NAD plus 2 ADP plus 2 pi, right? It gives 2 pyruvate plus 2 NADH plus 2 HTP plus 2 ATP plus 2 water, right? And next is the regulation of glycolysis. There are 3 major enzymes that control points within the glycolytic pathway. These include the hexokinase, phosphofructokinase, and pyruvate kinase reactions. This whole thing is actually a recap, so you remember whatever I've been lecturing on, and bioenergetics and the glycolysis pathway, the tri-acid cycle, TCA cycle, or the Krebs cycle, or the citric acid cycle, and all these things, gluconeogenesis, the entire thing. So these illustrations are going to help you a lot when you try to learn, right? And the 3 steps catalyzed by these enzymes are irreversible. Key drivers for regulating the pathway are energy demand within the cell as determined by local indicators such as ATP and AMP, as well as energy demand within the organism as a whole, which can be influenced by hormone signaling pathways. Regulation of glycolysis. Glycolysis is controlled or regulated by allosteric inhibition mechanism of 3 enzymes, as mentioned earlier. Hexokinase, it is allosterically inhibited by the product glucose 6-phosphate G6P in the process of negative feedback inhibition. The 20th PowerPoint presentation would be glycogen, which becomes glucose 1-phosphate and leads to the formation of hexosamine biosynthesis. And we have glucose 6-phosphate, which gives rise to hexosamine biosynthesis. In glycolysis, it gives rise to most cells, especially muscle and brain, to pyruvate and in the aerobic session. And the pentose phosphate pathway, it gives rise to ribose plus NADPH in most cells, and via pancreas it gives G6P, which is equal to sense of insulin release, and directly in the liver, through the liver, it gives glucose and then goes into the bloodstream. Cellular fates of glucose 6-phosphate, right? Cellular, just remember, telling you again, remember what is the cellular fate of glucose 6-phosphate. Remember this diagram, center diagram is highly illustrative, has a lot of answers in it. Glycogen becomes glucose 1-phosphate, which becomes glucose 6-phosphate, and conversions in various aspects. What happens with the fate of glucose 6-phosphate to all of these? Here we have this, and this. So 1, 2, 3, 4, and 5 effects, right? Remember that. The regulation of glycolysis PFK1. PFK1 is the most important control points in the glycolytic pathway. PFK is an allosteric enzyme and has a structure similar to that of hemoglobin. PFK1 undergoes complex allosteric regulation. 1/2 of each dimer contains the ADP dimer. It's dimer contains the ADP binding site, whereas the other half, the substrate fructose 6-phosphate or F6P binding site, as well as separate allosteric binding site that can bind with ADP or AMP. PFK1 activity is increased whenever the cell's ADP supply is depleted. PFK1 activity is decreased whenever the cell's ADP plus pi is accumulated. ATP and citrate 4-RMCCA acts as an inhibitor. Regulation of glycolysis through pyruvate kinase. Key regulatory component within the pathway. There are 3 major enzymes of pyruvate kinase: the L form that is predominantly found in the liver, the R form that is predominantly found in erythrocytes, and the M1 form in muscle and brain, and the M2 form that is expressed in fetal tissue and at some level in most adult tissues. You have general regulatory mechanisms common to most of the isozymes of pyruvate kinase. FPP is an early product within the same metabolic cascade. The activation of pyruvate kinase enzymes by FPP is known as feed-forward metabolism stimulation. All of the pyruvate kinase isozymes are inhibited by the product of the reaction ATP or high energy load and high levels of alanine. If high levels of alanine are present, this indicates that there is a high energy load within the cell, that is that the cell is full of building blocks to make new macromolecules and is not in the need of more energy. Thus, high levels of alanine serve as a negative regulator of the pyruvate kinase family of enzymes. Protein kinase A phosphorylates pyruvate kinase, inhibiting its activity and preventing the conversion of phosphoenol pyruvate to pyruvate. Now, the 24th power presentation point is regulation of glycolysis summary. Enzyme hexokinase, in the presence of no activator and inhibitor, glucose 6-phosphate increased concentration. PFK1 is the enzyme. Activator is fructose 2,6-biphosphate, fructose 6-biphosphate, AMP, increased concentration. Inhibitor is citrate, ATP, increased concentration. Pyruvate kinase, fructose 1,6-biphosphate, AMP is the activator. Acetyl-CoA, ATP, increased concentration is the inhibitor. Regulation of glycolysis in the pyruvate kinase. Here glycolysis, here is on the membrane, mitochondrial. It gives rise by difference, it's PK, it gives F2,6-PP, and then in the presence of PFK2 and FPPase2, it gives F6P, and then reversibly to F6P and F2,6-PP. And the presence of protein kinase A, it gives pyruvate kinase. In presence of phosphoenol pyruvate plus ADP, it gives pyruvate plus ATP. So glucagon signaling in liver cells downregulates PFK2 activity and pyruvate kinase activity. Right? Regulation of glycolysis in other steps. Step 6. Glyceraldehyde 3P dehydrogenase. For activity, reaction of the essential C-wise residues necessary by presence of heavy metals such as Hg2 irreversibly inhibits the enzyme. Glycolysis would soon come to a halt if the NADH concentration is increased in steps of 6 glycolysis. Substrate level phosphorylation. The formation of ATP by phosphoryl group transferase from a substrate such as 1,3-biphosphoglycerate is referred to as a substrate level phosphorylation. Substrate level phosphorylations involve soluble enzymes and chemical intermediates. The 28th PowerPoint would be role of NADPH in regulating the partitioning of glucose 6-phosphate between glycolysis and the pentose phosphate pathway, where glucose undergoes to become glucose 6-phosphate and undergoes glycolysis to release ADP. And the presence of the pentose phosphate pathway, it releases an NADPH and then becomes 6-phosphogluconolactone to release another NADPH and finally to pentose phosphates. Role of NADPH in regulating the partitioning glucose 6-phosphate between glycolysis and the pentose pathway. When NADPH is forming faster than it is being used for biosynthesis and glutathione reduction, NADPH rises and inhibits the first enzyme and the pentose phosphate pathway. As a result, more glucose 6-phosphate is available for glycolysis. This is another table for the entry of dietary glycogen, starch, disaccharides, and hexoses into the preparatory stage of glycolysis. You see this? Take a nice screenshot of this, right? See, endogenous glycogen starch. It's okay. It's the liver, the lactose part, the dietary glucose, starch, going to become glucose here to glucose 6-phosphate. Excuse me. Then going to fructose 6-phosphate here in the kidneys, the spleens, all these things. And next here in the liver you have fructose 1-phosphate, you have fructose 1,6-biphosphate, you have these finally glyceraldehyde 3-phosphate, right? You tap and the ADP, entry of dietary glycogen, starch, disaccharides, and hexoses into the preparatory state of glycolysis. The numbered stops are described, right? Here, glycolysis, you have glucose 6-phosphate. Sucrose becomes this, glycose, then it becomes fructose 6-phosphate. Sucrose and sorbitol convert fructose to, in the presence of fructokinase, to fructose 1-phosphate. Sucrose and sorbitol together form fructose. Here, in the presence of hexokinase, it becomes fructose 6-phosphate. And sucrose here becomes glucose 6-phosphate again. And then you have the conversions to fructose 2,6-biphosphate here. In the presence of fructokinase, you get fructose 1-phosphate. In the presence of PFK1, you get fructose 2,6-biphosphate. In the presence of aldolase and triose isomerase, you have glyceraldehyde 3-phosphate. In the presence of aldolase B, you have dihydroxyacetone phosphate and glyceraldehyde, which in the presence of triose isomerase gives glyceraldehyde 3-phosphate, which becomes pyruvate then. And glyceraldehyde in the presence of trikinase gives glyceraldehyde 3-phosphate, which again gives pyruvate. So it's alternate sugar sources for glycolysis. Time to find a solution. Calculate the standard free energy of the reaction catalyzed by the enzyme phosphogluconeogenesis. Glucose 1-phosphate irreversibly gives glucose 6-phosphate for the above reaction. Starting with 65 mm glucose 1-phosphate and no glucose 6-phosphate, the final equilibrium mixture at 25 degrees centigrade and pH 7 contains 2 mm glucose 1-phosphate and 63 mm glucose 6-phosphate. Thus, the reaction in the direction of glucose 6-phosphate formation proceed with the loss of energy. Time to find a solution. Calculate the standard energy change of the reaction catalyzed by the enzyme phosphogluconeogenesis. Glucose 1-phosphate reversibly gives glucose 6-phosphate for the above reaction. 90 mm glucose 1-phosphate and no glucose 6-phosphate. And final equilibrium mixture at 25 degrees centigrade and pH 7 contains 1 millimoles of glucose 1,6-phosphate and 89 millimoles of glucose 6-phosphate. Thus, the reaction in the direction of glucose 6-phosphate formation proceed with the loss or gain of energy. Yes. Glucose gives acetyl-CoA, which gives citrate and isocitrate with the release of NADH, gives alpha-KG, alpha-ketoglutarate, gives another NADH, then succinyl-CoA, then succinate, and the other side round it gives oxaloacetate, malate, fumarate, and then it goes on to release FADH2, O2, becomes water, ADP becomes AT, and the release of hydrogen ions is the 5 and 2, 3, 4, and 5. And this is the tricarboxylic acid cycle, TCA, or the citric acid cycle introduction. Okay, this is the answer to the above. Now we're going to deal with the tricarboxylic acid cycle, TCA, citric acid cycle introduction. Pyruvate, a node in the metabolism of carbohydrates, proteins, and fats. Anaerobic conditions, lactate in the cytosol, regenerating NAD+, for continued ATP production and glycolysis. Pyruvate has a diffusion capabilities, cytoplasm and mitochondrial chemometrics, and 2 steps: a diffusion through outer membrane pores, then via inner membrane carrier, symporter. By the carrier called H- coupled pyruvate specific symporter, the mitochondrial pyruvate carrier, pyruvate amino acid synthesis, or citric acid cycle for ADP and NADH energy, synthesis of fatty acids and sterols after getting converted to acetyl-CoA. The same tricarboxylic acid cycle, TCA, or citric acid cycle introduction. Pyruvate becomes acetyl-CoA plus CO2 by the enzyme pyruvate dehydrogenase, PDH complex. In PDH is 3 enzymes, 5 coenzymes, 4 cofactors derived from vitamins. Right? So the enzyme complex, PDH complex. Enzymes are E1, E2, and E3. And coenzyme prosthetic groups TPP, lipoid coenzyme A, CoA, SH, FAD, NAD. Cofactors are thiamine, pentosane, niacin, and riboflavin. Conversion of pyruvate to acetyl-CoA. At oxidative decarboxylation, note these diagrams, is an irreversible reaction oxidation process in which the carbox group is removed from pyruvate as a molecule of carbon dioxide, and the 2 remaining carbons become the acetyl group of acetyl-CoA. Starting material for TCA, NADH is produced, EDC. Conversion of pyruvate to acetyl-CoA. It's a pyruvate in the presence of pyruvate dehydrogenase complex E1, E2, and E3 gives E1, E2, and E3 is given here. Right? E1, E2, and E3. That's a pyruvate dehydrogenase, dihydrolipoyl, transacetylase, and dihydrolipoyl dehydrogenase. See, all these 3. And with the conversion of CoA, SH, and NAD plus to NADH in the presence of TPP and lipoid FAD gives acetyl-CoA. Overall reaction catalyzed by the pyruvate dehydrogenase complex. Conversion of pyruvate to acetyl-CoA. This is the diagram for that. Pyruvate dehydrogenase 1, dihydrolipoyl triacetylase E2, and dihydrolipoyl dehydrogenase E3. So 6 oxidative decarboxylation of pyruvate acetyl-CoA by the PDH complex. The path of pyruvate is traced in red in step 1 pyruvate. TCA cycle, Krebs cycle, citric acid cycle. First cyclic pathway, just note the numbers here in the diagram, and then you relate with this. Oxidation of acetyl-CoA, citric 6C produces compound, 8 different steps, 3 reversible steps, key regulatory steps, 3 NADH, 1 FADH2, and 1 GDP or ATP is produced by, and pyruvate via acetyl-CoA entering the cycle. No net removal oxalate occurs. NADH and FADH donate the electrons to the respiratory chain where electron flows direct drives ADP synthesis. German chemist Hans Adolf Krebs is the one who proponent of this. Condensation, Krebs acid cycle, oxidative decarboxylation, and then you have the substrate level phosphorylation, and you have the cycle where fumarate converts it to malate, fumarate to malate, T-malate to oxaloacetate, regulation of TG cycle, stringent regulation to balance the supply of key intermediates with demand of energy production, biosynthesis, regulation at steps, oxidation of pyruvate to acetyl-CoA catalyzed by PDH complex, inhibited allosterically by ADP and acetyl-CoA and NADH via phosphorylation of PDH. And when ATP, ADP, etc., all of which indicate an energy sufficient metabolic state. Regulation of TCA, citrate synthetase, isocitrate dehydrogenase, and alpha-ketone glutarate. Take a screenshot of each of these. Here, high radiation, all these dehydrogenase, the reactions is through mass action. Dehydrogenase reaction is essentially at equilibrium in the cell. Product accumulation inhibits all three steps. Sectional CoA inhibition, alpha ketoglutarate dehydrogenase and citrate. And ATP inhibits both citrate synthesis and isocitrate dehydrogenase. Regulation of TCA. Calcium 2 plus the signal for contraction and for concomitant increase in demand for ATP activates both isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase as well as the PDH complex. The concentration of citrate, the product of the first step of the citric acid cycle and an important allosteric inhibitor of fructokinase 1 and of the glycolytic pathway. Dichloroacetate DCA inhibits PDH kinase in the laboratory and so relieves the inhibition of PDH complex, DCA, and apoptosis. Bioenergetics of glycolysis and TCA cycle. Glucose to glucose 6-phosphate 1 ATP and number of ATP ultimately formed. Okay, here it's -1, here it's -1. I hope it's right. Fructose 6-phosphate to fructose 6-biphosphate. So this entire table is noted down. TCA cycles anaplerotic reaction. Here we have reaction pyruvate plus HCO2 plus ATP, which gives oxaloacetate plus ADP plus F2 and liver and kidney. Second one is in the heart and skeletal muscle. Third is in the high plants yeast bacteria, and fourth is in the widely distributed eukaryotes and bacteria. TCA cycles anaplerotic reaction. Here is a screenshot for that. Take a note of that. Citric acid cycle serves in both catabolic and anabolic processes, and some interesting info. These points 4 and 5 carbon intermediates. The cycle serve as precursors for a wide variety of products. To replace intermediates removed for this purpose, cells employ anaplerotic replenishing reactions. All the enzymes are in the matrix of mitochondria except succinate dehydrogenase, succinyl CoA synthetase, or succinyl thiokinase, indicates the participation of a nucleoside triphosphate in the reaction. Then in anaerobic organisms, the citric acid cycle is an anabolic pathway. Then you have the oxaloacetate and alpha ketoglutarate precursors of aspartate and glutamate by simple transamination, purine and pyrimidine nucleoside synthesis. Succinyl CoA central intermediate in the synthesis of porphyrin ring of heme group, which serve as oxygen carriers in hemoglobin, myoglobin, and electron carriers in cytochromes. Oxaloacetate can be converted to glucose by gluconeogenesis. Conversion of acetate of acetyl CoA to glucose occurs in bacteria, plants, fungi, and protists. The glyoxylate path cycle, metabolons, supermolecular assembly of sequential metabolic enzymes, example malodehydrogenase, citrate synthetase, and aconitase metabolism. Citrate acid cycle activity changes in tumors, downregulation of mitochondrial pyruvate carrier, MPC, and PDH. Leads to pyruvate accumulation of cytosol, accumulation of lactate and succinate, oncometabolites, leading to tumor growth. Acting through specific G protein coupled receptors, GPCRs. Mutation in citric acid cycle enzymes, succinate dehydrogenase, lead to tumors of the adrenal gland. Mutations in the fumarase gene lead to tumors of smooth muscle layer, myomas, and kidney. Questions to work out. Net equation for glycolysis in the citric acid cycle. Write the net biochemical equation for the metabolism of a molecule of glucose by glycolysis and the citric acid cycle, including all cofactors. Thiamine deficiency, formation of oxaloacetate in a mitochondrion, and role of a vitamin thiamine, right? Electron-Transport chain and oxidative phosphorylation. Oxidative phosphorylation, mitochondria with the help of protein. The first one you have is the culmination of energy-yielding metabolism, catabolism, and aerobic organisms. Mitochondria with the help of protein complexes embedded in the inner mitochondrial membrane. Electron flow from electron donors oxidizable substrates to a chain of membrane-bound carriers to a final electron acceptor with a rare large reduction potential. The final acceptor is molecular oxygen O2. The free oxygen made available down an hexagonic electron flow is coupled as the to the uphill transfer of protons across a proton-impermeable membrane. Transmembrane flow of protons back down the electrochemical gradients through specific protein channels provides a free energy for synthesis of ADP. Chemiosmotic theory by Peter Mitchell 61. It is a movement of ions across selective membrane, permeable membrane down the electrochemical gradient leading to formation of ATP, electron-transport chain oxidative phosphorylation. So depiction of the production of ATP using the process of chemiosmosis in mitochondria is called oxidative phosphorylation. Electron-transport chain and oxidative phosphorylation. Take a screenshot of this. Electron-transport chain oxidative phosphorylation. Types of electron transfers occur in oxidative phosphorylation. Right? 1, 2, 3 transfers, 3 other types. Ubiquinone. So I've given all these details in my formal lecture. I'm just giving you an illustration, so just take note of each, okay? You can just magnify it, see the whole thing. That's the diagram for the electron transport chain in OP. Complex name, nature, component, and subunits and work. Complex is 1, complex 2, complex 3, cytochrome 2, cytochrome C, complex 4, complex 4, cytochrome oxidase every 4 electron passes through this complex. The enzyme consumes 4 substrate H form the matrix side in converting O2 and to H2O. Q cycle proposed by Mitchell. Take a note of these. Alright. Electrons reach Q through complex 1 and 2. Reduce Q, QH2 serves as mobile carrier electrons and protons. Passes electrons to complex 3. Complex 4 then transfers electrons for O2, forming a reduced cytochrome C. Electron flow through complexes 1, 3, and 4 is accompanied by proton efflux from the matrix intermediate membrane space. Chemiosmotic coupling of ATP synthesis to electron transfer by transfer from different charge and pH. Inhibitors of ATC and ATP synthesis. Complex name inhibitors. Inhibitors of ATC and ATP synthesis. Metformin, fenformin, retinol YM146, 3-ANPA, melanate, TDFA, antimycin A, cyanide, opigamycin, G-box. Proton motive force. Free energy change for the creation of electrochemical gradient by an ion form base alpha G, delta G is equal to RG, C1, C2, C1 by C2 plus ZF delta psi, C1 concentration. C2 and C1 are the concentrations of iron in 2 regions. Note these points. ATP synthesis by ATP synthase. Chemiosmotic model. The representation on the figure ADP plus PI plus NHP giving ADP plus 2 plus NH. Free type ADP is complex Y is peripheral membrane protein. ATP synthesis, ATP synthase. First 3 beta and alpha subunits, 1 delta. I think that's a smaller form of delta unit, right? Central shaft, gamma subunit, lambda subunit. Gamma, lambda, this is gamma subunit. Alpha subunit, oligomycin sensitivity, FOS or C-ring, made up of number of C-rings, small hydrophobic proteins, provides path for proton movement. Paul Boyer proposed rotational catalyst mechanism, ATP synthase. Binding change model of ATP synthase. Regulation mechanism. In hypoxic oxygen deprived cells and nicotine beta blocks ADP hydrolysis, but the reverse ADP synthesis preventing a drastic reaction. That adaptive response is hypoxia. To hypoxia, ATP and ADP concentrations set at the rate of electron transfer. Additional information. Proteins with cytochromes are proteins with characteristics. The strong absorption of visible light due to their non-iron cytochromes designated A and B, types A cytochrome, B cytochromes, C cytochromes. Additional information. Iron sulfur protein, one of a large family of electron transfer protein in which electron carriers one or more ions associated with 2 or more sulfur atoms of C residues or of inorganic sulfide. Take a screenshot of these. Riesque iron sulfur protein, type of iron sulfur protein in which 2 of the ligands to the near central ion or ion are S side chain. Nucleosides, nucleotides, nucleic acids, structure, diversity, function. These are the photographs of the famous scientists, Francis Crick and James Watson, and there's someone, this Frederick, and these are the nucleosides, nucleotides, nucleic acids, structure, diversity, and functions. So all the details are there. Two types of nucleic acid, DNA and RNA. Nitrogen is base linked, made up of nucleotides. Nucleoside nitrogenous base linked IC. Nucleoside nucleotide. It's nucleotide nitrogenous containing nice base, pentose sugar, one or more phosphate group. Nitrogenous nitrogen containing base, planar aromatic heterocyclic molecules. Two types, purine and pyrimidine. Purines is heterocyclic aromatic organic compound consisting of pyrimidine ring fused to an imidazole ring. Two types, adenine 6-amino purine, guanosine in a 6-oxy-2-amino purine. Nitrogen containing base, cytosine uracil thymine. Sugar types, deoxyribose DNA, ribose DNA, beta-D-ribose, beta-deoxyribose, 5-carbon atom, stable structure. Nucleotide and nucleic acid nomenclature. Adenine, guanine, pyrimidines, cytosine, thymine, uracil. Nitrogen bases, the DNA found in RNA and DNA, right? Adenine, guanine, cytosine, uracil, thymine, ribonucleotide, ribonucleoside, nucleoside, nucleotide, deoxyribonucleotide, nucleoside, and nucleotide. Phosphodiester bonds link 6 nucleotides, nucleic acids. Diversity of RNA and the function RNA. And nucleic acid, messenger RNA. Ribonucleic acid, types A, mRNA, messenger RNA. So all points to be noted, okay? Take a screenshot. And types based on sedimentary rate types. Diversity of RNA and the function tRNA, trans-RNA. Same. Ribonucleic acid types, tRNA, trans-RNA. The crystal structures of tRNA are the important points. Functions. Structure of DNA, major form of B form. Structure of DNA, major form of B form. Structure of DNA, major form of B form. These are all extensive notes on that. Note this diagram. And chart graphs. Rule is pure and pyrimidine base pairs are in equal amount. A plus G equal to T plus C. A plus G by T plus C equal to 1. Other types of DNA, helix, RH, LH. Right? Understanding the flexibility of the sugar phosphate backbone. Nucleic acids are insoluble in water. When attached to pentose sugar and phosphate to become nuclear soluble. The hydrophobic phase of the bases displays a strong constraint on the overall conformation of large DNA or RNA molecule in solution. The solution phosphate backbone of the polynucleotide. Sugar pucker refers to the conformation of ribose or deoxyribose. Sugar conformation defines how the nucleotides are linked to each other in space. The sugar pucker is defined by the position C2 and C3 atoms related to a plane formed by the C1, O4, and C4 atoms. The sugar puckers in DNA RNA structures are predominantly in either C3 endo, aDNA, RNA, or C2 endo. Importance. Storage of genetic information, important to Watson and Crick's proposal, replication, inheritance, suppression of genetic material. And comment when Watson and Crick really took from Franklin what they took. Photophosphorylation, non-cyclic phosphorylation, non-cyclic photophosphorylation, cyclic photophosphorylation, right? And these are the references. So that's it about this chapter. We just closing on the illustrative aspects of bioenergetics related procedures, right? So please make adequate screenshots and learn the terminologies and the cycles, very specific for both the exams, for the AIIMS, for the NEET biology, and for the, even of course for the All-India PG, and for the CSIR-NET exams of the University Grants Commission. Learn all these and you should be in good stead to get good marks, right? So work on all that, you should be fine. Best wishes, and we go to the next chapter soon.